So we're starting the final presentations, and this will allow, there'll be 15 minutes presentation and then 5 minutes of questions. So the microphone for questions thereafter is kind of the name of the game for any of the final project oral reports. Lisa DeVetter will be presenting on optimizing nutrient management for organically grown blueberries east of the Cascade Range.
All right. Well, good morning. For those of you that weren't at the table I sat at for the Speed Network situation, I'm Lisa DeVetter. I lead the Small Fruit Horticulture program at Washington State University. I'm based in Mount Vernon, which is in the western part of Washington State, but this research is occurring in a really important production area about 4 hours away from where I'm situated, depending on traffic in Seattle, in Eastern Washington. So the title of this presentation is Optimizing Nutrient Management in Organically Grown Blueberries East of the Cascade Range. And this project has been funded not just for 3 years, but for 6 years. So I'm actually going to be going over 6 years of data that we have from funding with the Northwest Washington I forgot the acronym, Northwest Center for Small Fruits Research, as well as funding from the Washington Blueberry Commission. I'd like to acknowledge these many individuals who helped contribute to this research, particularly given it was several hours away from where I'm located. And then there's several other individuals that I'll acknowledge later on. So a little bit about the background and need. So Eastern Washington is a leader in organic blueberry production. Just in 2023, Washington State produced about 40% of the total national supply of organic blueberries. And most of that production is concentrated east of the Cascade Range. Now, despite the scale of this industry, we have very little guidelines on organic nutrient management. Now, there's been some excellent research done here in Western Oregon, led by the late Dr. Bernadine Strick, but there's a lot of questions about the translation of that information to that unique production area, where we have differences in soil characteristics. For example, pH tends to be much higher. Organic matter is a lot lower. We have differences in climate. Plant productivity also tends to be greater. So a lot of the growers were under the assumption that the nutrient requirements would be greater to account for the higher nutrient removal of that crop. And then the fertilizer sources were different as well. So as I mentioned, there's been a great history of organic nutrient management research in blueberry here in Western Washington, and I wanted to highlight some of that here. One of the takeaways from that work by Dr. Bernadine Strick and her collaborators was that there's cultivar responses. Liberty was less sensitive to fertilizer source and rate compared to Duke, which is one of our main cultivars and the one I'll be focusing on for this presentation. And Duke performed better with feather meal as opposed to fish emulsion, which is one of the main nutrients that we use. Also, there were higher yields in Duke with low nitrogen fertilizer rates as opposed to higher rates. But again, a lot of the growers in that particular production area were asking, do these results and subsequent guidelines extend to our unique production area? So that brings us to the objectives. So the objectives were to evaluate the effects of commercially available organic nitrogen fertilizer sources applied at low, medium, and high rates on yield, fruit quality, plant and soil nutrients in blueberry east of the Cascade Range. And we were focusing on Benton County, which is where most of the organic production is concentrated. We also looked at the dynamics of nitrogen in the fertilizer sources in this particular study. And then lastly, we worked to disseminate project information. So a little bit about our approach. So we started this trial in 2018 in a fourth leaf Duke field in Prosser, Washington. It was a commercial field and it was organically managed. The experimental design was a split plot in a randomized complete block, and we had 4 replications for a total of 48 plots. Each plot had 12 plants, and so it was a pretty huge trial. We had 4 fertilizer sources at 3 different nitrogen rates that we were evaluating. So a little bit more on those fertilizer treatments. We had 4 sources applied at 3 different rates. All of this was decided upon with grower input And so our sources were Wise Organic, blood meal, True Fish Emulsion, and a combination. The Wise Organic, if you're unfamiliar with it, it's derived from digested plant material, and it's a liquid just like the fish emulsion. And our rates are 50, 100, and 150 pounds per acre of total nitrogen based on the fertilizer analysis. We applied the fertilizer around the base of the plants, and this field was established with weed mat, and it was on a raised bed as well. We also applied supplemental fertilizer, specifically phosphorus and potassium, in 2018 and 2020, so that it was equivalent across our treatments, but we discontinued doing this after 2020, and I'll point out why we did that. It's an important part of this research. So some of the data that we collected, we looked at yield and fruit quality. We also looked at leaf tissue nutrients, and we spent a lot of time looking at various soil characteristics. For example, we looked at soil nutrients, and we used these plant root simulator probes, which are ion exchange membranes that look at ammonium and nitrate in the soil. And we also looked at soil temperature and moisture. And I'm just going to be presenting on a subset of these data given the time. So let's look at yield. So we have, if you're on my right, I'll be pointing here. We have yield per bush on our y-axis, and I have this divided by year because we saw some interesting trends over the years. And then on the x-axis, we have our various fertilizer source and rate treatments. And then just a tip, as you're following along, this orangish color is our blood meal fertilizer source. Green is our combination. This teal color is our fish emulsion, and then in purple is our Wise Organic. So it was complex. There were a lot of three-way and two-way interactions. So three-way interactions with fertilizer source, rate, and year, and then two-way interactions with our source and rate. Overall, in 2021, we started to see declines in yield, and I attribute this to the fact that the grower cooperators put a tunnel over their field, and that really limited the honeybees' ability to detect polarized light, forage, and pollinate those bushes. And we saw that throughout the duration when they had that tunnel. But overall, when we looked at the data, yield was largely unaffected by treatments in most years of the study. We did see effects in 2018 and 2021. And in 2018, just focusing on that year, yield was greatest among plants treated with the fish emulsion and lowest for Wise Organic across all rates. But I'm a little suspect of this. I don't have a good explanation why, but this was the first year of the trial. So with a perennial plant, I would expect that some of these fertilizer effects would take time to accumulate. But again, little suspect because we didn't see that trend later on in the trial. We did see another response in 2021. Plants treated with the high rate yielded more than the low rate across all sources. However, even though it wasn't significant, yield tended to be lower in our blood meal and our combination treatment, which also had blood meal in it. And I suspect that the way that we were delivering the blood meal on the surface of the soil didn't necessarily allow that nitrogen to work its way down through the root system. Now let's look at fruit quality. So this is a busy table. Let's walk you through it. We have our treatments here, both our source of fertilizer and the rate, and berry weight, total soluble solids, pH, and titratable acidity. When it comes to berry weight and our total soluble solids, we only saw a difference by year, not by any of our treatments. For pH and titratable acidity, there were interactions, and you can see, if you're curious and looking at the p-values, they're there. But overall, when we look at the data more closely, what we saw that berries tended to be more acidic, both in terms of pH and the titratable acidity, at the low rates and less affected by the fertilizer source. And then 2023, I don't have the data all analyzed, but I'm expecting it's going to follow the same trend as we saw the previous 5 years, where we saw a year effect, but not effect due to any of our fertilizer treatments. Now tissue nutrients. So here we're looking at total nitrogen, phosphorus, potassium, and calcium. We looked at other nutrients as well, but the interest of time, I'm going to be focusing on these nutrients. And here at the bottom, I want to point out too, we have the sufficiency standards for Oregon and Eastern Washington. So we saw many interactions again, but overall, leaf tissue nitrogen was highest at the high rate across all sources. Phosphorus really only differed by year, and then potassium was highest at our high nitrogen rates, and calcium the converse. It was highest in our low rates. And what I suspect was happening here is that we had some ion competition, that the higher the rates of the potassium, it reduced the uptake of the calcium, and we saw that reflected in our leaf tissue data. All of these nutrients, with the exception of potassium, were within the acceptable ranges. Let's look at the leaf tissue nitrogen and potassium data a little bit more closely. Here we're looking at leaf tissue nitrogen on our y-axis, our different fertilizer treatments, and here I have the data broken down by year so we can look at trends over time. And then right here in orange, this is our Oregon tissue sufficiency standards, and then our Washington tissue sufficiency standards. So overall, in most years of the trial, our fertilizer treatments were in the sufficiency standards, with the exception of blood meal and the combo at the low rate in some years. And again, that might be attributed to that blood meal and the nitrogen within not working its way fully into the root zone. Potassium. So this figure is set up similarly. We've got our Oregon state standards and our Washington state standards. So very clearly you can see that we often exceeded the sufficiency levels. We did see a decline when we discontinued the application of the supplemental phosphorus and potassium, specifically in this case. So that's encouraging to know. If you're a producer and you see your leaf potassium going up beyond the sufficiency ranges, stopping that potassium really quickly leads to a decline in those levels. We also noticed leaf potassium was highest in the Wise Organic combo and lowest in the blood meal. And this is one of the recommendations coming from the trial. It's not new. Other researchers have found this as well. Use caution when using fertilizers containing leaf or potassium, because that high leaf potassium is negatively correlated with yield. Now for the soil component of the data. So here again, we're looking at pH, electrical conductivity, and just soil potassium. We looked at other variables as well, but in the interest of time, we'll focus on this. And then we have our recommended ranges. So we saw no fertilizer source effects for these data. pH overall was lowest at our high fertilizer rate and highest at our low fertilizer rate, which again is pretty consistent with previous trials that have been done on organic nutrients. And then our electroconductivity and potassium was highest at the high rates, which can also make sense. A lot of these organic fertilizers are high in salts as well as high in potassium. Now, objective 2, this was the work done by my collaborator, Dr. Gabe LaHoo. What he did is he looked at the nitrogen mineralization of our fertilizer sources. So just to take a moment to get oriented, we have the amount of inorganic nitrogen being released from our fertilizer sources over the course of a 112-day incubation trial in the lab. And our temperature treatments were 55 and 65 degrees Fahrenheit, which are representative of the soil temperatures that blueberry roots in this production region experience when they would be taking up fertilizer nitrogen. And then we have our treatments, and if you notice, we have a control, and this is a treatment where we was coming from a natural field that had no history of fertilizer application. So the goal here is really looking at what's being released by the soil and the organic matter in the background. So overall, what Dr. LaHoo concludes is that fish emulsion, blood meal, and the combination treatment have the highest inorganic nitrogen, with Wise Organic being intermediate between those sources and the unfertilizer control. The rate differences were mostly attributed to that initial inorganic nitrogen content in our fertilizer sources and its net mineralization within the first 14 to 28 days. So those fertilizers are releasing the nutrients relatively rapidly. And then the nitrogen mineralization from the organic fertilizers, that initial rate response, well, he concluded with that initial rate response within the first 28 days was largely due to the fertilizer. And then after that, it's largely a background soil organic matter. Now, objective 3, so that's information dissemination. What we've done so far, we have 1 trade journal publication and 1 scientific publication. We also have had multiple conferences and field days, and some of this information has been also incorporated into a pomology course that I teach at Washington State University and some of our Master Gardener training classes. Of course, we want to finish the data analysis and scale up and take on continued extension efforts for this project and other great projects that have been happening in the region with regards to nutrient management in both conventional and organic systems for blueberry. So next steps, we want to finish analyzing our PRS data. We're still waiting on the 2023 data from the lab, as well as the firmness data. I think it's also really important, and the survey reminded us how important it is to contextualize this information with regards to economics, because these fertilizers vary in costs. And then we also want to look at an additional scientific publication. And then last but not least, work with some of the excellent scientists here at Oregon State University and USDA to update the Pacific Northwest or nutrient management for blueberries guide and have it more representative of production areas across the entire Pacific Northwest. So some of our conclusions from this trial, what we learned or what we're concluding is that established Duke is tolerant to a range of organic nitrogen fertilizer sources. Now, if you recall, some of the initial work by Dr. Burning-Strick found that the feather meal was more favorable for Duke, but we didn't test that because what we heard from our growers is that that's largely unavailable for them. So this is just for the products that we tested in Duke. Mineralization of the organic fertilizers occurs rapidly within the first 14 to 28 days, according to the incubation trials. And our recommendation moving forward is an annual rate of approximately 50 pounds per acre. So the low rate is a good foundation to build a fertilizer program around. We do have some words of caution moving forward. So be careful with the blood meal. We had some concerns that that was, again, working its way into the soil. We were applying it similarly to our grower cooperator was applying it. And of course, adjusting it based on the crop load and on tissue nutrient analyses that are done annually. We also recommend using caution with fertilizers containing high amounts of potassium, because again, as those leaf potassium levels build, that can be associated with negative, lower yields. And low fertilizer rates have some benefits. Number one, it can reduce the fertilizer cost as well as buildup of soil electroconductivity, potassium, and potentially other nutrients in that fertilizer source that can lead to nutrient imbalances. And what we're seeing is that lower rate can help maintain yield or fruit quality overall, and leaf sufficiency levels for nitrogen. And then, a little bit harder to extrapolate on, and we haven't measured this, but at these lower rates, you have potentially less loss of nitrogen to the environment through volatilization and leaching. I do want to point out too, because I think that's pretty surprising that the low rates performed as good as the medium and high rates. This is consistent with the work in Oregon as well as work elsewhere, that the lower rates seem to work just as well, if not better, than some of the medium or high rates. So I want to acknowledge not just our grower cooperator, but the many people that helped out with this project, from collecting yield data, soil samples, etc. Some of these other organizations that helped donate fertilizers. Dr. Burning-Strick, who was very helpful in providing guidance as a resource for this project, and again, funding from the Northwest Center for Small Fruits and the Washington Blueberry Commission. And with that, I would be happy to take any questions. Oh, Devin, yes.
So, hi, I'm Devin Rickner. Great presentation, that was awesome. Something I didn't see over time, did you see EC build up in those soils over time? Because that EC value is getting pretty high.
Yes, so Devin mentioned about the EC values getting high. We did look at it over time, and yes, over time it was increasing. Yep. Yeah, Dave.
So you noticed the first year that you had higher yields.
Mm-hmm.
Wondering if perhaps you also showed that the mineralization is a bit higher due to lower temperature fish than the other fertilizers. Not significant, but what Verity found was, the fish releases nitrogen very quickly. In fact, in her feather meal treatment, she used that to jumpstart the plant. So I'm wondering if perhaps the plants were nitrogen deficient in that field. So consequently, that first year, they got a boost from the fish fertilizer. And then after that, then the other fertilizers kind of caught up and things started to level up. Do you think that's what happened?
Yeah, no, I appreciate the comment from Dave. I maybe can repeat that if people didn't hear on the other end of the room. So Dr. Breila's comment was, well, previous work shows that fish emulsion releases a lot of nitrogen, so it might be helpful kind of jump-starting these younger plants, and then the other fertilizer treatments could kind of catch up and even things out. That is a possibility, kind of looking at some of Dr. LaHoo's data with the nitrogen release. We do get a bigger boost and a higher rate, so that's one of the considerations that we're thinking about. So thank you for that. Yeah, great. All right. Great. Thanks again.
University. And the title of my presentation today is Exploring. Hi, good afternoon. My name is Dr. Jim Harbertson from Washington State University. And the title of my presentation today is Exploring the Interactive Effects of Grape Maturity, Alcohol Concentration, and Long-Term Maceration on Wine Sensorial Properties. So first off, this project was a combined effort of funding, if you will. And initially, the winemaking project was funded by the Washington Wine Grape Funds. That project is a much bigger one for 3 years, and in it that project was designed to evaluate the impacts of ripening in pre and post fermentation alcohol adjustment on wine chemical composition and sensory attributes. For this portion of the project, we just carved off one of the winemaking years, which was funded by the Washington Wine Grape Funds, and we did a slightly different variation on the initial experiment. So for the objectives for the Northwest Small Fruit Project, we were to do sensory evaluation of the wines made from that project, and also a chemical evaluation of the proanthocyanidins, otherwise known as tannins. So what is the project? The project is a very large one. In this project, we are doing 3 different harvests, and they are consecutive harvests, and each one is done to target a particular soluble solids or bricks. And for each one, we've also adjusted to control for alcohol, because we know how alcohol has an effect on extraction, and it also has an effect on perception of aroma and things. So we've essentially targeted for a low sugar concentration. In this case, we've adjusted it back down to 20 from 22, and we also then adjusted it to 24 and 28, which were the other targets. So these are all done in triplicate. On top of that, the wrinkle on the original project from Washington Wine Grape Funds was we added an additional long-term extended maceration project. Now, this is Cabernet Sauvignon fruit, and a large part of what winemakers want to understand about them is about astringency changes as a result of harvesting at different times and then winemaking techniques. So this project is really designed to look at the interaction between both of them. And you'll notice that these are very long-term macerations, so 240 days. Remember that our tanks at the Wine Science Center are about 220 liters, so they're almost about the size of a barrel. So this is kind of the wrong size for doing a red wine fermentation so it's very short, small surface area, and mostly large volume comparatively. So it's not so extreme as it would be in industry, although this does get practice from time to time. So in terms of doing the sensory analysis, we did descriptive analysis, which is pretty standard for doing sensory of wines. We had 11 trained panelists. We did 16 aroma attributes, which were chosen by the panel consensus. 3 taste attributes and 3 mouthfeel sensations were also evaluated. Because we didn't want to overwhelm our panelists, we did we only did 2 of the winemaking replicates, because it's essentially which were done in triplicate, as opposed to all the winemaking replicates, which would add too much work to them. And we did these evaluations in booth with black glasses and red light due to the fact that these are red wines. And because they're also very astringent wines, we enforced a 3-minute time between samples where they were given crackers and sparkling water so they could rinse. So finishing this commentary, all of the attributes that were evaluated, both aroma, mouthfeel, et cetera, these were all done on a linear scale, all being done with the CompuSense software system from Canada. So what were the aroma attributes that ended up being significant? So we had 8 significant aroma attributes: light fruit, dry fruit, vegetables, chemical, alcohol, cheesy, yeasty thing, nutty, and overall aroma, and 3 taste descriptors: bitter, sour, sweet, and the three mouthfeel sensations: astringency, hot, and viscosity. So these attributes were all come up with by consensus by the panelists, and these were found to be significant to analysis of variance after we did the sensory evaluation. So what does this sensory tell us? Essentially, we've attempted to boil all of that data down for you, because there'd be a lot of graphs if I showed you each analysis of variance for all the different attributes. So this is a graph showing you being done by principal component analysis. And if you're kind of familiar with these, hopefully this is just a review for you. Essentially, this is a modeling mathematical technique that allows you to sort of evaluate all of the sensory attributes all in one go, and then sort of place those samples into a space so that you can understand how your different treatments varied by all the various different sensory attributes that your panelists have rated. So in this case, this is the loadings plot on here on the left, showing you that we have these long vectors, which is astringency, dry fruit, bitter, nutty, alcohol, chemical. Over on this source portion of it, we have light fruit, sour on this portion, vegetables down here, cheesy, rancid, yeasty, all that kind of components. On the other side, I have these confidence ellipses of the mean points for the various different wines. So I'll just sort of show you how they're being labeled so you can kind of follow along. So on this section, as an example, all of these here are essentially 28 Brix wines that have been treated. So this is a 28 Brix here. This is the control of 28. And then we've chaptalized here, we've taken the 24 Brix fruit that's been harvested earlier and then chaptalized up to 28. And this one down here, the 20 to 28, is fruit that was picked at 20 Brix and then chaptalized up to 28, if you will. So you can see kind of already that the idea that they might actually be aligning based on alcohol as opposed to being when they were picked per se. And more or less, when you kind of put sort of these little ovals around them to sort of show you the groupings, this is not about significance, it's just showing you kind of where they fit into the grand scheme of things. You can see that the wines more or less separate based on when they were picked and also some of the treatments that were done. Essentially, the wines that were made with much higher alcohol are all associated on this right-hand side of this plot. All the 28s over here, all of the nutty, alcohol, chemical, aroma, intensity, sweet, that kind of business. And then on the other side, where you have the 20 Brix wines, if you will, the sour, the cheesy, rancid, yeast, vegetable kind of attributes are kind of connected to those ones. And then we go further than that, you'll find that the wines where that have undergone extended maceration are also all being pushed upwards up into the upper quadrant, indicating that they are more astringent, more bitter, also tend to have more of this dry fruit characteristic, potentially a little bit of nutty and things of that nature. So overall sensory results. Essentially, the alcohol and extended maceration treatments separated the wines distinctly for aroma, mouthfeel, and taste attributes. The veggie aromas were essentially diminished in the higher alcohol wines, as well as wines made from more ripe fruit. We've seen this before. Alcohol has a tendency to sort of mask the perception of vegetative flavors. This is one of the better examples in Cabernet Sauvignon. Results also indicate that harvest time was essentially less important than the alcohol concentration and maceration on the sensory results. This was a little bit of a surprise to us, given that this is Cabernet Sauvignon, but we've also seen this in other examples in Syrah and Merlot. So essentially, these results suggest that pre-fermentation amelioration, whether it be juice runoff and water addition or chaptalization, are effective tools to alter wine sensory profiles and can be used to mimic earlier or later harvest wine styles, meaning that you can pick early, add a little sugar, end up with a wine that seems a little bit riper, or likewise take something that's a little bit riper, water it back a little bit, and then you also get a little bit less ripe kind of characteristics in the wine. So now we've moved on to the section of our work that was to do with measuring the tannins. So my group in Washington is well known for measuring protein precipitable tannins. You can kind of see that we do get what you would expect, a lot more tannin when you do the extended maceration. So in the examples here, you have the 20 Brix where you've got a control and extended maceration. You can see that there's a slight uptick with the higher alcohol in the initial winemaking stages, and this is diminished later on as the wines are harvested later and later. So you kind of see, although there aren't very obvious differences in some cases, like this one over here, this is not much different than that one. But there is a trend, if you will, as the wines being made later, having a little less protein precipitable tannin and being sensed, in fact, as being slightly less astringent. So the protein precipitable tannins actually correlate very, very strongly, but a 0.99 correlation with our data for the stringency within this particular experiment. So the protein precipitable tannins were very good at essentially predicting how astringent the wines were going to be without having to really do the sensory, if you will, just measure the protein precipitable tannins. Now, we wanted to actually learn a lot more about the tannins per se, as opposed to just being able to just say that they're protein precipitable, because there's a lot that's changing in the fruit of the grape with tannins. Now, I had a slide dedicated to showing you all the chicken wire and the tannin structures and everything. I like to show that thing, but suffice to say, I think that slide is a little bit too complicated for the amount of time that we've got here. So I'm just going to summarize what we've done. So we pulled apart the tannins, looked at the composition of them, the size of them based on analysis known as fluoroglucose analysis, something made famous by Dr. James Kennedy, who was a former faculty member at Oregon State University. So in this case, we've used his method and said that wines made from 28 Brix fruit had larger tannins with a greater proportion of subunits derived from skins and fewer from the seeds. About a 40% increase from the 22 Brix fruit, but not different than the 25 Brix fruit. The results are consistent with skin tannins being larger than seeds, but not necessarily the fruit data. In the fruit data, we found that it indicated that the skin tannin maximum size was about 25 Brix. We also found that the wines made from the extended maceration had tannins that were 50% smaller than the controls. This is something I've seen before in my other studies. And we also found that those tannins were primarily derived from the seeds, which is why they were much smaller. And there's a greater proportion of seed-derived monomers as well. The grape skins have no monomers in them, no bitter things, only astringent things. And so when you do extended maceration, you tend to emphasize getting more of the monomer and much more of the smaller polymers coming out of the grape seeds. So in terms of an industry significance, the results of this study show that longer ripening in Cabernet Sauvignon makes wine that proportionally have less seed and more skin-based proanthocyanins. The study also demonstrates that pre-fermentation amelioration, whether it be juice runoff and water addition or chaptalization, are effective tools to alter wine sensory profiles and can be used to mimic earlier or later harvests. In terms of what we are seeing as changes to standard production practices and new winemaker recommendations as opposed to grower recommendations, right? Here's winemakers in this case. Winemakers are interested in limiting tannin and catechin extraction from grape seeds from Cabernet Sauvignon are recommended to extend grape ripening and avoid the winemaking technique extended maceration. The study here shows very clearly that you can avoid that by just picking later, just kind of nice. Winemakers can also mitigate the impacts of climate change by using chaptalization or juice runoff and water addition to mimic wines made from earlier or later harvest, depending on what the weather is going to throw at you. We've seen so many different things happen up in Washington the last few years, whether it be too hot or too cold or anything. It's nice to know that you have winemaking tools that can be used to sort of adjust for the style of wines that you want to make. So I have some acknowledgements to make. I did the presentation, but the students do the work. This is Juliana Pazos and Danny Fox. And the money for the original project to make the wines came from the Washington Wine Grape Funds. This is from our Management of Phenolic Compounds and Vineyard and Winery Investigation, Mechanical Pruning, Grape Maturity. So the wines were made by that project and not funded by Northwest Small Fruits. Sensory and the chemistry was funded by Northwest Small Fruits. So I also have to thank Chateau Ste. Michelle Wine Estates for giving us the fruit to do this experiment. That's a lot of fruit to do this experiment from the Coal Creek Vineyard. Former winemaker Matt Bonsley, who's now moved on, and now the current winemaker Tino Valdez. And then also on the original grant was a postdoc who moved on, did not actually participate in this project, but he helped plan some of the sensory, Scott Frost. And so if you want to get a hold of me, I have my email right here for you, jfharbertson@wsu.edu. If you have any questions, I can tell you right now. I know those of you who are working in Oregon, Willamette Valley, are probably primarily working with Pinot Noir. A lot of what I've shown here is possibly not entirely applicable to what you do, but I am open to doing projects with Pinot Noir. So if you're very interested, let me know. With that, I will say goodbye.
Okay, next up we have Inga Zasada presenting on risk assessment of nematode-transmitted viruses of small fruit crops in the Pacific Northwest.
All right, hello again. I was thinking about what Julie said, difficulty of growers and stakeholders understanding the relevance of what you're doing. So I'm going to just start with the relevance. Nematodes everywhere, virus not. So keep on doing what you're doing, which is planting clean plant material. That really is the foundation for protecting our crops from nematode-transmitted viruses. Start off by acknowledging my co-authors. So this was a project that was funded in 2020. Hired a postdoc, Lori Reinhold, on this project. She kind of suffered through COVID with us, and then she got a job at NCGR. So she's currently the curator of several of the collections at NCGR. Had to hire someone else, and we're very fortunate to find Christian, who is in the audience here. He joined us in 2022 and kept this project moving forward. Oops, excuse me. So what we're talking about here is a disease complex. We have viruses, and we have what they're called nepoviruses. So they're nematode-transmitted viruses with polyhedral particles. Christian has a bunch of photos from our survey effort in 2023 showing the various symptoms that you'll see when you're out in small fruit fields or vineyards of what these type of viruses look like. On the other side are dagger nematodes within the genus and species, Ziphinema americanum, very complex group of nematodes that also at very high densities also have a direct impact, especially on vine growth. And that's shown here in the pictures. One of the vineyards we went to down towards Medford, short and internodes, just really struggling vines. We can also see down on the bottom, patchy distribution of plants in a field where this disease complex exists. And again, the relevance is the virus is quite devastating. Once a nematode-transmitted virus is found in a small fruit field or a vineyard, plant death is going to occur over either a very rapid pace or just over a couple years. And unfortunately, I can't say that for nematodes, but for viruses, yes. So a little bit complicated table here, but just wanted to go back and look at what we know about this virus nematode complex. Here we list the different genera of viruses, the viruses that nematodes will transmit, the various hosts, and then the vector. And the vector is the nematode. And essentially what we want to show you is that there's a tremendous diversity in viruses. There's a tremendous diversity in the type of dagger nematodes that are going to transmit these viruses. And there's a tremendous diversity in the hosts. So our project did encompass all of the small fruits that are grown in the region to try to get a handle on this. And the ones that are underlined, tobacco ring spot and tomato ring spot, are the viruses that we were interested in this study. A few things to point out here. Second one down, grapevine fan leaf virus. This is a big deal in California. Luckily, it's only been found once in the Pacific Northwest, but never in association with its vector. So that's good news for us. And then second from the bottom, cherry rasp leaf virus is quite widespread in the cherry producing orchards of the Pacific Northwest. So what do we know? From a nematode perspective, Jack Pinkerton and Paul Schreiner have done a lot of work in this area. First characterized nematodes found in Oregon vineyards in the 1990s. We furthered that effort with early surveys in wine grapes in the early 20s. We've also done surveys in raspberries and blueberries in the region. But most stuff has happened more than a decade ago. So kind of the justification to take a second look at this, especially with a lot of chatter that I was hearing from various people. So this kind of shows what we knew before Christian, Lori, and I started down this path. And the viruses, different types of viruses are in colors. Tomato ring spot virus is TORSV, and tobacco ring spot virus is TRSV. We then have grape family virus, and then two other viruses that I can't remember what they are because I'm not a virologist. And then Ziphinema americanum and where it is present. So first of all, you can see nematodes are widespread or have been reported to be widespread in the region. And we kind of have a smattering of occurrence of these different viruses across the region. Again, nematodes widespread, virus is not. So what were the objectives of our research to kind of build off of what we already knew? So the first thing was to get an updated assessment of what was going on. So conduct a survey of nematode transmitted viruses and nematodes in Pacific Northwest small fruits. And then second, a lot of the previous research that was done in this area was before the advent of all this really cool advanced genomics technology. So really we wanted to start to bring technology to bear to what I consider is probably a very difficult disease complex to work with. So objective 1, our survey. COVID slowed us down quite a bit on this project, unfortunately. But Lori went out in 2021 and did the initial survey effort. That was mostly in wine grapes in Washington and also small fruit fields in the Willamette Valley. And then Christian went out in 2023 and we finished off the survey by going to Northern Washington, looking at raspberries and blueberries, and then going to Southern Oregon and considering wine grapes down there. And so we went around, we collected plant material, soil samples, and took all this back to the lab and analyzed it. In total, we visited 96 different small fruit fields in the region to try to get a really good grasp about what was going on. Here are our results real quickly. Again, it's the message I told you at the beginning. So if you go to the very bottom, so again, these are the crops that we looked at, the number of samples that we had. On the bottom are our totals. And I just want to focus on the bolded numbers. 52% incidence of dagger nematodes in the region. We expected this. But again, very small incidence of tomato and tobacco ring spot virus in the region. Just showing down there, average dagger nematode densities, 22 nematodes per 250 grams of soil. That's about normal for what we see in the region. If you're super unlucky, you'll have 530. So again, for folks who are looking at nematode reports, anything over about 50 with dagger nematode kind of causes me a little bit of alarm. Not tremendous alarm, but just it make me kind of scratch my head and think maybe I should do something. This is an updated map of the areas that we sampled. And what I really want to point out here is that we kind of expanded our knowledge of where these viruses are. And that was super important to us. And I'll show that to you in the next part of the talk. But we were able to, unfortunately, detect tomato ring spot virus in several locations up and down the I-5 corridor. And we also are probably looking at the first report of tobacco ring spot virus in wine grapes in Southern Oregon. So moving on to our second objective is to develop sensitive and specific molecular tools for identification of nematodes and viruses. And this is really important to push this field forward. You look at the literature, and probably the last time that anybody really dove into this topic was 20 years ago. So we have a long way to go to kind of get tools up and running to be able to ask some really cool questions. And it was more difficult than we thought. COVID made it difficult. But just kind of the diversity of what's going on in the region also kind of made it more difficult. We discovered that we don't know a lot about the diversity of nematode transmitted viruses. And when I talk about diversity, I mean their genomes. They're quite complex and they can vary from isolate to isolate. We realized that it's hard to work in a system where you don't have nematodes that have virus already in them to be able to really do experiments. So we needed to find those sites where the nematode and the virus were both present. And then finally, again, we needed dagger nematodes that had the Nepo virus because otherwise it's really difficult to understand what's going on. So for each one of these questions, the things that we needed to push this research forward, first of all, we must know the diversity of tomato ring spot virus for primer design. And this came about because, like I said, in our survey, we were collecting plant material, bringing it back to the lab, extracting RNA, and then running ELISA or standard primers that are used quite commonly in the industry. And Christian started to notice that these primers that he was pulling out of the literature and testing against this material, they weren't all doing the same job. So that's shown on the positive out of total. So ELISA, 32 isolates out of 33 tested positive with ELISA, but you start to look at this 7 different PCR primers that Christian was trying to use, and you can see that they vary greatly in their ability to detect tomato ring spot virus. So this was a little concerning to us. And the way that Christian decided to try to get around this was to do high throughput sequencing of the isolates that we were finding so that we can essentially develop robust, inclusive primers that we fill with 100% confidence we're actually detecting the virus. So that was one of the little hiccups in the road that we had, and this is still ongoing, and Christian has made a lot of progress towards finalizing this part of the project. Second, we need to know the presence of the virus and the vector in small fruit crops in the Pacific Northwest to find hotspots. So we were able to achieve this through our survey effort. We have data from 2021 that Lori collected, data from 2023 that Christian collected. Our first go around, we found nematodes in quite a few places, but no virus. We had a few more leads in 2023. We heard from people that there were certain places we should go. So when we went out, we were very lucky. We found a bunch of locations, a bunch meaning 6, locations where we had both the virus and the nematodes. So this sets the stage for us to be able to go back to these locations, collect nematodes, collect material, and be able to piece together this complex a little bit better. And then finally, we need Dagger nematodes harboring the Nipah viruses. And the way that we're trying to get at this, and this is exceedingly difficult, and I commend Christian for all his work on this because it's not easy, is trying to work in an experimental setting with Dagger nematodes. They literally will drop dead if you look at them. And so you have to have a lot of reps. You have to, be really patient and create a experimental framework that's conducive for these nematodes. And Christian has gone a long way to getting us there. And on the left-hand side, you can see our greenhouse cultures. We probably have the only Dagger nematode culture in the world, to be quite honest. So, you need to have a source of the nematodes. You need to treat them nicely as you put them through these small pot experiments. You add plants that have virus, hope the nematodes pick up the virus. You can then find the virus in the nematode. It becomes quite complicated, but we certainly are getting to the point where Christian has a really good system. On the other hand, can we go to those fields where we know where the virus and the nematodes occur and just go out to nature and be able to collect nematodes and kind of fast track this whole system? So that's what he's also done there, collecting nematodes from these hotspots. We can use cucumber plants. Naidu's done a lot of work in this area. Stick a cucumber in. It's also a host for the virus, and you can start to understand if the nematode has the virus and is able to transmit it. So we continue to work on that, try to finalize these methodologies so we can take the next step to kind of teasing apart this complex. So in conclusion, again, Dagger nematodes are widely prevalent. Luckily, the viruses that they transmit are not. Again, clean plant material obtained from certified nurseries is the best way to protect yourself. We know that tomato ring spot virus diversity is broad, and we also know that some of our current primers do not detect all isolates. And then finally, population densities of Dagger nematodes are variable, but often surpass proposed threshold levels, and that's in cases where we find the nematode in the field. Ongoing work. Christian will continue to validate the RT-qPCR for tomato ring spot detection in nematodes. We can go back to our locations from the 2023 survey to be able to have nematodes and virus to work with. And on our side of things, Amy Peets is here in the audience, lab manager. We're working on mitogenomics, so sequencing nematode mitochondrial genomes to be able to come up with robust markers to identify these nematodes. And with that, that's the end. A lot of people helped us. If you allowed us to come onto your farm or vineyard, thank you so much. A lot of people hooked us up with people to contact, and thanks to the Northwest Center for funding this effort. And I'll take any questions. Julie?
So I noticed that in Washington, you did find some nematodes, but very, very little in the way of virus. Oregon was the flip.
Correct.
Should this really be forefront of the radar for the Oregon wine industry?
I would still say no. And that's good news, right? Because again, the number of places that we found the virus was very small. And to be quite honest, I think most of the sites where we found virus in Oregon vineyards, we had a very hard time in finding the nematodes. So that means it came in on dirty plant material and it probably won't move from that spot. Die.
A lot of our plantings went in after other crops in Oregon. So the plum orchards, good sites. So we had probably nematodes there. It's nice to know that the virus.
Yeah, exactly.
How do you sample? Do you use like probes?
Yeah, we use a probe down to 12 to 18 inches and just collect multiple samples and then composite them. Yeah. 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 Yeah, for sure. And so the question was about the movement of dagger nematodes in the soil profile and when's the best time to sample for them. I think it depends on where you are. So Jack Pinkerton always talked about sampling in the spring in Oregon to detect dagger nematode. I believe that is true because you have moisture in the soil profile. I think in the summer they move, because the soil dries out a lot unless you're irrigated. So I would recommend spring for Oregon. But for Washington, with that really defined irrigation zone, I think any time of the year is good for sampling for dagger. For blueberries, yeah. If you're on irrigation, I don't think the nematode is going to try to move away from a non-conducive environment. Yeah. Yeah.
What's the persistence of the virus in
Mm-hmm.
the replant situation? How long?
So how long will the virus persist in the nematode? It depends on if the nematode has a host to feed on. So the nematode will lose the virus every time it molts, and nematodes molt 5 times. So in a field wall where there's an alternate host and the nematode's completing its life cycle, it will shed the virus. And if the virus is no longer there, it won't reacquire it. So, but if you're in a fallow situation and you have good weed control, I think that the nematode and the virus probably could persist for a year or so. And some of Jack's work back in raspberry, back with Bob Martin, kind of demonstrated that longevity of virus and nematode in a raspberry system. Yeah.
Is there any relationship between nematode and virus? And for example, specific virus group only infected this nematode or vice versa?
Yeah.
Did you find any?
So is there a specific relationship between certain groups of viruses and nematodes? And there's a bunch of virologists in this audience. It makes me nervous. But I think historically you look at the literature and it's a very defined group of viruses that nematodes are able to pick up. And then the nematode's very specific as well. Yeah. Right? Yeah.
Who came to the sites that you had
There were a few folks that were experiencing some issues that came to other people in the unit. That was one way to get clued into it. And then our contacts, when we asked them to come up with a list of vineyards or small fruit fields to go, we usually ask for areas that maybe were experiencing a little decline. So that kind of led us in that direction as well. Yeah. Great. All right. Thank you.
Okay, that's all for the morning oral presentations. Lunches will be in the foyer, and for those attending the wine grape and raspberry blackberry breakout sessions, those will be in ballrooms A and B at 12:10.
So to be clear, wine and any grape-related thing will be in ballroom A. Berries and small fruits or berries and specialty crops will be in ballroom B. Thanks, Mike. Gotcha. Okay. There, right there. Check that there. Should be good. Yeah, thank you.
We went last year. We were in the same table last year. Yeah, I remember.
The way I look at it is, that's the reason you do the work. I know you have to publish and all of that, but the satisfaction of it. You know, and the community, and I think through that work. Yeah. Yeah, we all need that input from time to time, because you know, our tendency is to see everything within our little world and not. We're gonna have some bad tips. We're not achieving that ultimate success, and because that and the process. When you're flooded out, it's individuals and sometimes who knows what we do, but the more of those transfers that we happen through those individuals, the higher you're gonna actually get out there sufficiently.
Very strong. Making sure you resource
But this even. I think that's. But to get. Yeah. Figure out how to do it. That's the challenge. Like we get
What happened?
It's like a.
She was like, that was the only time.
Yeah, it's not. Yeah, she did, last time she was like, did pick up the phone and now.
Against cancers of.
Yeah.
Needed.
Yeah, it's so sensitive, it seems like. Yeah. Like. So if you are having a share that start coming up and it's not expressing their thesis expression. Yeah. Yeah, it's.
Here's video of this. You know, like, yeah, I don't know.
No, I know. Seriously. Yeah.
So once we have the method,
have the concern or having any much of it in your field. We need to be testing that on the right if it's very the ability of design if it's make a or the virus is not present. It's not the first person.
Yeah. Because it's a little bit responsible.
Question on that.
It's not taking
Or doesn't it?
Yeah.
We have that. But really, it's those alternatives down there.
Yeah, I want to go to the other one too.
Yeah. So,
Yeah.
how that get bumped and helps.
No, it's-
It's still that. Yeah. Lot of-
Yeah.
lot of- one, I can't think of, passing arts, primers. And is that right?
All the sequences, the virus genomes and see the virus
That's
genomes.
the whole primer thing is pretty fuzzy in my mind. That some sort of a material that brings out something that the computer helps the computer to see.
You can think, for example, about COVID.
Okay.
And when you have the text, then the COVID part. But many times the test came negative besides we were expecting. That is because that text, that variant that you are covering. So what we are doing is developing a small sequence that oriented a small sequence that will allow us to detect all these variants.
The piece of just 150 ATGCs that are either in the 150 pieces. And to tell you what it is, there's this primers that just, that a couple just lays down and allows you to just look at those 150 letters.
What happens is a filter.
Yeah,
source.
yes, exactly.
Focus on what you want to
Exactly
see.
what you want. I think the future, probably, whatever the end is, the whole genomes, the machine, but right now, I think that.
Right now, they are raised to that not investigate. So that's why our work is trying to inform on kind of field applicability or bigger scale application, right? Just more research, researchers can handle the small chunks of. But is right. So you can use the in Oregon or in this right. And in Willamette, in the
Diagnostic. They start government methods. Minus primers, minus government. And that in itself is just like, well, why isn't there?
Got another question. So in your lab, you develop primers. You realize your value of that, something that's used in other labs.
That's.
Is there a way to, because there's a cost to develop this, right?
Yeah.
Just a goodwill
Goodwill
thing.
thing, yeah.
We're all in this together
Yeah,
somehow.
and for me, I think at the end, I be a key for it. Yeah. So,
It's not something patent.
no, yeah. Some people know, primers and
License
yeah.
this use.
Especially in
Yeah.
Korea, they have
Okay.
yeah, in Korea, they can have a new people using all over the face. And our thinking is, anyways, they funded that. And it's coming growth, it's something of our taxpayers. So it's not a mostly they can do make a work use that taxpayers money. But for them, something that they think that is they do. That is why we are trying to find that doesn't make it. Of course, we need papers, but yes, at the end it will be more, you know, maybe the word or like be more better.
Yeah, more satisfied.
More, yes, that's it.
Yeah.
It's more satisfying that you see that someone is having having a benefit.
I look at how much the Rexburg industry has changed over the last 15 years, from moving to education, poly on beds. I mean, people, it's nice to see, right? It makes you realize that, that a little bit that you did towards, making people comfortable with doing these changes as well. Yeah, so, it's so different. I had been up for years and was driving around. I didn't know if I've ever experienced an industry kind of change so much horticulturally. Just the way that something is run.
Based on its establishment.
Yeah, yes, on the establishment.
Yeah.
Yeah, it's something else.
Yeah. It's a big value that's been being realized by utilizing that technology or like that. Well, it gives you the edge. It's like it's challenging to make it work for a long time. So we've got to improve where reasonably can. New ideas, which cost, there's a cost to that, and there's the risk cost, and all of that, and then you mitigate some of that through research or comfort, and it really just takes testing it or utilizing it at a scale that would communicate enough confidence that, this is reasonable, this is doable, and then Lisa's then follow up to generate the data on the difference. It's like, it's a no-brainer, right? Really, even though there's still some of the growers that it's too intimidating because they don't have the machinery, they haven't done it before, so they're going to stick with not going.
But as they drive around, and you start to see the percentage of acres that's going to switch to.
Oh, yes. Yeah, it's the majority of acres that are planted now are under plastic. How to manage that, of course, is, it's important to get comfortable with, laying it out, retrieving when the house is really going to work, move it on. What about my nutrient program? Put that on there, you need to think it through, and the more research that is, developed to give it information about what you really need to do, or this isn't that important have it removed and they get back to a more traditional nutrition system. No, they're seeing really good results like that. But then again, I mean, they're using some genetic material too. It's really colchicine in grisicles.
Oh, I haven't heard about that.
That is the go-to, that is the choice. It is the way cave and Wakefields are kind of has-beens. That's all that's changing. So they are selling those plants as fast as they can.
So whoever's your house is, really?
Let's do bigger.
Let's do bigger.
Yeah.
Yeah.
Most of it is just field growing. Colchicine is the variety of what the earlier one they had was vodka. Well, I don't know if they really commercialized it. They were just, this is John and Martin here. Have been working with Driscoll for almost 20 years. Their material, and so this is the latest one, and it looks really good. I think it just really looks good. Now, it's only been out, and being utilized for 5 or 6 years. Serious acreage. And, you know, cropping, bring up cropping systems, things start showing up over time that can be problematic for them. Seems to be quite early. But yes, that's I think an unknown. And over time, that will become, you know, time will elucidate so the weaknesses.
Yeah, you know, in Wakefield, you know, I sampled a lot of Wakefield, you know, when it first came out. And it was the best post fermentation. I mean, always able to maintain this ginormous root system. And that root system on that thing was just like,
all the time.
yeah, you put a meter and.
Where are the roots?
Yeah, seriously, where are they?
Live.
I know.
Compare it.
Yeah, and it was
The
just,
root mass difference.
oh, it's
you know.
huge. Yeah.
I know. And I think that's probably,
This family's got it.
it sounds like it. I know that graders kind of weird though. They're first. They're also had planted in Haven. But not with success. Haven. But the root development difference between Colchicine and Wake Haven is striking. So I think a lot of it has to do with root levels and how, you know, aggressive the root systems are regenerate.
I agree, it's like, whatever.
Exactly.
Roots, you know.
Yeah, feeding, you know, nutrient,
Oh, yeah, all of
you
it.
know, accessibility, it's mining soil and.
That's
Yeah,
what
you
about
know,
Wakefield. Well, and Wakefield, I literally dig down this far, just a massive amounts of this. I always wondered with Wakefield, that persisted for their soil profile, because it was just super small.
I just wonder about the root microbial interactions and that, you know, over time in support of more diversity and higher populations of beneficial bacteria, fungi in that rhizosphere. Or maybe it's more of a magnet for, you know, parasitic.
And yeah, and I think in Wakefield it is, because you know, a lot of nice fine feeders, but the plant's ability to produce new roots is inherent, even when there are crap ton of nitrates. Because there were some of those Wakefield lips, right, like 10,000 nematodes, you know, it's just like,
doesn't seem to
How's this
matter a
going?
whole lot.
Yeah, surviving, right?
Yeah, it's really
Yeah.
interesting.
And again, you know, as a researcher, the only way you learn those things is by going out in the field and seeing it. Because, you know,
You've been so good at that.
Yeah.
Over the years.
yeah, and it's just like,
You've been such a joy to work. I mean, as a scientist and also now, the last 4 or 5 years. wanted to share something with you that I forgot to talk about. I'm finding it kind of hard to reach my daily caloric intake, considering I'm eating such, low, caloric density food.
Yeah.
Do you have to eat so much more of it to reach my caloric intake?
Yeah.
And I think the reason why I get like tired and stuff like that is I think like I'm not reaching, I'm eating bad stuff, and I'm not necessarily reaching my caloric intake, and I'm backloading my food intake to about the time that processes in my system. Like it feels like my system hasn't eaten anything still. So I think that's why I feel tired and lethargic. An observation I have.
Well, the other thing too, and you really, do they ever talk to you and stuff? I mean, eating 3 meals a day and not.
I've been eating bananas.
Okay. Well, and also with the being lethargic too, if you're not getting, good exercise, that's gonna make you lethargic.
I'm just saying, trying to eat like the same amount of calories in which I actually burn is kind of getting hard.
Okay.
I'm just saying.
I don't know what to tell you. Do they have
I'm not looking for advice, I'm just telling.
Oh, you're just telling me. Okay. Are you being plateaued on your weight?
I went up and then I went back down. Let's see. I think it was that not like late as in like after 5. Like I said, I'm backloaded.
Yeah.
Or yeah, backloaded. Okay. So weighed in at 263.8. 262.8 this morning. That was 265 yesterday. 265.8 the day before. Then 263.2, 263.4, or 263.4 before.
So you kind of plateaued, you lost 3 pounds, and then you kind of plateaued, which is normal.
Yeah.
That's pretty normal, lose 3 pounds, plateau there, and you're going to hold there for, a while.
What's crazy is they predict that I will be at like 253 by Christmas, which is just, I cannot envision it.
Well, you keep following, their plan.
I know, I'm just saying, I can't admit, like, I scroll to like Easter time.
Yeah.
2:30, it's supposed to be 2:30.
That would be great.
Mm-hmm.
That would be really, really great.
I'll reach my goal weight on June 23rd.
That is at 200.
That's 200.
Okay.
So I'll be in summer beach shape on it.
Okay, the holidays are gonna be a challenge.
I know.
Well, I'm even thinking about that as far as, I'm not gonna make as much food for Thanksgiving.
Well, I'll try not to puke on Christmas Day. That should.
Do you think we can talk to Joe about, I said, Becky started a new program called Noon. And actually, she knows somebody who's doing that too.
Yeah.
And is liking it.
Well, the thing is like, as long as I don't, because what happened that day, if you remember right, I was unconsciously eating that whole day.
Yeah.
how was it, you know? And that has stopped me a lot of stuff because usually like when we have like a phase out for events and stuff, I'll just grab it, or I'll grab something, you know, grab some cheese, just, you know, grace. But like, since I am recording my food, I usually, you know, I have to think about it, right? I have to think, stop and think about eating, because then that means I have to be conscious about it, because I have to then, you know, I have to then think of it. So it becomes from my unconscious to my conscious, and also it's like, do I really want to have to go in the app and record this? You know, like,
No, this is really great, Joe. This is what you needed.
this is just like the rocket, this is rocket money for my weight.
Yeah, that is, that's what I'm saying, you gotta find, in the future, you gotta find, you know, clean your house something.
Yeah.
So that you respond really well to this kind of.
Structure.
Structure, you do, because it's very objective, it's not, you know, me in your head, or Andy in your head, or anybody else in your, it's a very objective deal.
Yeah.
That is just really helpful to you.
It's like almost somebody like, Joe, you need to do this, Joe, this is what you, if I have a picture of like what I need to do, if somebody gives me a list of things to do, or like instructions on what I need to do, and how I need to do, I'll nail those. Like, because I have a structure. Like if you leave me to my own, you know, devices as far as something I know, I have a very hard time structuring my own life. You know, I'm very creative and very, you know, spontaneous. I'm very like, I'm able to roll with the punches very well. That's some of my strengths. But some of those strengths are also my weaknesses as far as not being able to structure myself and all that kind of stuff. So.
Well, just hang in there, keep going. I think you know, plateau, but hopefully, if you keep with the program, you're gonna start, you know, you're gonna start getting off that 163, and you'll be dropping the next one. You'll go under, you'll go 162 something. You'll go, oh, cool. You know, you might bounce back up, but pretty soon you're back to, you know what I mean? It just,
all right.
Yeah, so that's good.
I am.
Did you contact Tim and apologize?
Oh, I didn't want that. So that night, I said, Tim, listen, I apologize for not being back to you. I really don't want this to ruin what was an amazing experience. I was already in a weird place and stuff. Love you, I'm happy with it. And it kind of bit me, but he returned and said, apology accepted. Hope your mental gets better. Which I was like, just let it go, you know.
Yeah.
I was just like, all right. But then today, he texted me, he's like, hey, I want to apologize for how I reacted the other day. I should have considered anything when it comes to inviting someone to connect. I was taught a different way of doing that. Anyways, love you.
Oh, that's good, right there.
Yeah, and I said, love you, Mom, Tim, which is an inside joke for us.
Yeah.
And he said, I hope you're having a good day.
Okay. See, you guys, sometimes when you take the high road like that, like you did, you humble yourself, then it really gives the other person a chance to reflect. And that's what Tim did. He reflected, and that makes me feel better about Tim. Maybe you guys have to learn this too.
Well, I think probably too, he talked to Nikki about it. And I don't think Nikki necessarily has the same views on that, but.
Yeah. So, okay, all was good. Especially since he initiated that second.
Yeah.
That exchange, that was good.
Yeah.
All right, good. But
No, I didn't change
in the
anything.
future, if somehow he finds some money.
And that run like.
Right away.
Right.
You know, yeah. Don't even, if you tell me something you're gonna do, I'll say. All right, okay. You get, have a good day.
All right.
All right, bye-bye.
Early back.